Hi, everyone. Thanks so much for joining me today. A big welcome to the Allogene Therapeutics team. My name is Wintana, and I will be showing you all how BioRender can help support you in communicating the important research that is being done at Allogene Therapeutics. So the agenda for today is we'll just go through a brief overview of BioRender, and then we'll walk through some design techniques and some functionalities of BioRender. Since this isn't a live demo, feel free to send any questions that you may have to our support email, as well as my personal email. And also feel free to check out our frequently asked questions, as it is super useful. So a bit about me. My name is Wintana Siyum. I am a customer success associate here at BioRender. I studied biology for health sciences at the University of Toronto with a stream in molecular biology. And for those of you who don't know, BioRender is a web-based platform for creating scientific images, but it's been designed with scientists and researchers in mind. So this means that, we have simple functionalities such as dragging and dropping, meaning you don't need to have a design background to use BioRender. At the end of the day, our goal is to give every scientist the tools and practical knowledge to visually communicate their research. So on that note, let's jump right into the tutorial. So now we are in the gallery view of BioRender, and we can start our illustration in two ways. So the first is starting off with a blank illustration, or we can use a template. And a template are images and texts that have been combined to obviously explain a process. And these are nice because we can edit them to our liking. They are extremely customizable. But for the purpose of today's demo, we will start with a blank illustration. So we are in the Canvas view right now. And what I'll do is just hide the little video at the corner so nothing is being blocked. Okay, perfect. So you'll see that on the top left, we can actually change the title of our illustration. So I can call it Allogene. Perfect. So let's take a tour of what exactly we are seeing here. On the left-hand side, I like to think of this as a binder with different tabs. So each tab has its own set of sections. So as you can see, the icons tab, you'll be happy to know, has over 35,000 icons, and all of the icons are customized and have been categorized into a wide-ranging library, which is great. And you'll see that no matter what tab I am on, the search bar at the top always remains. So I can type in something specific and relevant to my illustration. So let's say I'm typing in a cell, and anything relevant to that word will just pop up in the middle here. And another trick I like is a saying I like to say is hover to discover. So I'll hover my mouse over an icon and a description will pop up as to what that icon is. So this is a petri dish with cells, and this is a stem cell. So once I am happy with the image I see, I'll just utilize that drag and drop functionality and put it into our canvas. At the very top here, you'll notice what we like to call a toolbar. So this toolbar is great because it is dynamic. It changes according to whatever we have selected. So the default is our ability to just insert lines and shapes. However, if, for example, I select this image, I can now change the transparency, or I can arrange the order, or flip it vertically or horizontally. So it's nice. It's a nice tool to have because it adjusts to, again, whatever you have selected within your canvas. On the very right, we have some super useful navigational tools. And then on the bottom right, we have some helpful resources such as our live chat, our frequently asked questions, and some keyboard shortcuts. So let's say we're creating a diagram that provides a basic outline of how CAR T cells are developed and its effect on cancer cells, to be specific. So what we'll do, first I want to show, for example, how the T cells and an inactive virus combine to create the CAR T cells. So I'll type in T cells here, and I'll hover to discover and see that this is a T cell with the TRCs, which is exactly what I want. And then I'll type in a virus to represent that inactive virus that, contains the genetic code for the chimeric antigen receptor. And since this doesn't have like a DNA in the middle, I want to be a little bit more specific. So I'll type in DNA and drag and drop that right in the middle here. And what's nice is with any image, I can just go ahead and resize it according to my liking. So right now I'm just going to go through and put in any relevant images for my diagram. So I'm going to type in a CAR T cell, drag and drop that because that'll be the next part of my diagram. And then I'm going to go into my favorites tab, which I'll cover later on, and drag and drop this image that I've created. And I'll show you how to do that soon. But this is just showing the cancer cells and the CAR T cells combining to ultimately create our dead or dying cancer cells. Lovely. So now I have, the basic outline of my diagram, and I did that with simply dragging and dropping. So let's talk about the difference between bounding boxes, because I think that's super useful to know. So you'll see that, for example, some icons are surrounded by a purple bounding box and others are surrounded by a blue bounding box. So blue bounding box means that this is a set icon and we aren't really able to change specific or individual parameters of the icon. So I can't make this node bigger than this node. It's just not something that we can do. However, for anything covered with a purple bounding box, we can make individual changes because these are grouped icons that have been clustered together to create this image. So for example, I can double click and highlight this receptor and I can change its color. I can resize it. So essentially I can make changes to this individual image without affecting the whole of the icon, which is great. So basically a blue versus a purple bounding box hints to the level of edits we can make to each icon. The third tool I want to explain while I change this back is the difference between locking and grouping icons. So let's say I'm super happy with the placement of these two images. I would highlight both of them. And to know that both images are highlighted, you'll see that both of the bounding boxes have appeared. And I'll go to our dynamic toolbox at the top and I'll press lock object. So what does that mean? You'll see that if I'm trying to drag or move these images, they are locked into place. So this function is super great. For example, if I have an illustration with a lot of images and I want to work on one area without making changes to another, so I can just lock it in place. And if I do, for example, want to unlock it and make those changes, I can, no problem. So the next thing I would like to cover is grouping icons. So this is what I was alluding to before. If you recall, these two were separate images. So what that means is if, for example, I want to move this virus over here, the DNA doesn't come along with it. But no worries, BioRender has come up with a feature called grouping. So again, I'll use that highlighting functionality. And both images are highlighted. And at the top, I'll press group. And now you'll see it is covered in a purple bounding box because these are grouped icons. And now we've created our own little image, which is nice. And if I'm obsessed with this image and I'm thinking, hey, like I don't want to keep creating it, no worries. All I have to do is highlight it and click favorites. And now it is saved into our favorites tab. So for example, as you can see, I have a lot of different versions of it. So I can just keep, dragging and dropping to my liking, but I've created my own icon. So let's say I wanted to add some arrows to show or provide some direction to our diagram. So I would go under arrows and I'll choose this curved arrow because I want to show that the T cell as well as the virus are being combined to reprogram the T cell. So we'll just add that there. And then I can change the width of the lines. There's a lot of different functions that I can play with in terms of the arrows. And then I can add another arrow between this and this step, showing that once the CAR T cell comes in contact with the cancer, we have dead cancer cells. So other things that we can do is insert some text box to provide some context or label. Virus. and so on and so forth. And of course, if this wasn't a demo, I would definitely provide more detail. But for the purpose of the tutorial, once we want to see like how everything will look, we can press preview illustration. And this is how everything will come out. So we can edit accordingly. So I could adjust these arrows or make this bigger or smaller. But let's say I was happy with my image. I can go in, press file, save, and there we go. Just in a matter of minutes, we created a quick and easy diagram, and we've demonstrated the underlying biological process that allogene therapeutics is seeking to revolutionize. So within this tutorial, we learned dragging and dropping techniques, blue versus purple bounding box. We learned how to lock or group icons, save icons, and we can even implement arrows and text boxes. So thank you again to everyone at Allogene Therapeutics for joining me. If you do have any questions, again, feel free to message our support email as well as my personal email. I am really excited to see the innovative ways you all utilize BioRender to illustrate your research. So thanks so much, everyone, and have a productive week. Bye-bye.